field crops educator with MSU Extension down in the southwest corner of the state, and I will be one of your hosts this morning. Now, hopefully you've taken the opportunity to do a speaker check and your system is working fine. If you do have any technical questions during the program, please contact the help desk at 844-678-6200. You can communicate with us a few different ways this morning. At the bottom of your screen, you will see a chat and a Q&A button. Chat is for making general comments or responding to questions from the presenters. The Q&A button is where you can type in your questions that we will present to the speakers. We'll also have a few polling questions throughout the morning. When those appear on the screen, just click on the appropriate button to participate. For those of you who are joining us today on your phone with voice only, you can still ask questions. Just send a text to Megan Honke at 989-666-3773, and we'll include them with the others. So write that down. That number again is 989-666-3773. Megan works with our events management team, and she will be in the background today, making sure that things run smoothly. Megan has been the key to bringing this virtual field day to you this morning. We're glad you're able to join us today, and want you to know that all MSU Extension outreach programs are made available to everyone in the general public. And I want to take a moment to thank the Corn Marketing Program of Michigan for their ongoing support and for funding a portion of the research you'll hear about today. I would like to introduce Kristen Poley. She's the research manager with Corn Marketing Program and the Soil Health Partnership, which is a national corn growers initiative. Kristen will be moderating our field day and introducing our presenters. I'll be helping to monitor the chat in the background throughout the morning, and I'll make sure that your questions are addressed. With that, Kristen, thank you for joining us today. I will stop sharing my screen and I'll turn things over to you.
Thank you, Eric. Welcome, everyone, to the MSU Corn Virtual Field Day. Again, my name is Kristen Foley, and I'm the research manager for the Corn Marketing Program of Michigan, which is your corn checkoff program. Michigan Corn is committed to funding initiatives that reflect the interests of the corn industry and that will return significant value to our corn farmers. In case you missed it, we just wrapped up our 2020 Between the Rows tour virtually this year, where we shared agronomy and marketing updates, as well as our estimation of corn yields for this year. If you would like any information on anything covered in that program, please reach out to me. Michigan Corn is proud to have a long-standing partnership with MSU and MSU Extension, and we are happy to be part of today's program. Without further delay, I will introduce the presenters for our first session. The first half of the morning will be focused on a relatively new disease in Michigan, tar spot in corn. We have a number of experts at Michigan State University, including Dr. Marty Chilvers, the field crops pathologist, and his graduate students, Emily Rogenkamp and Jill Check, who will discuss their research into this new disease and management tactics. Next, Dr. Addie Thompson, a corn geneticist, along with her graduate student, Blake Tragstad, will discuss their research looking for plant resistance to tar spot. Lastly, Lyndon Kelly and Youngsok Dong will talk about the importance of managing leaf wetness with this disease and how fungicides can be applied through overhead irrigation. After the discussion on tar spot, we will dive into our second session focused on managing uneven corn stands and hybrid selection. If you have any questions, please type them into the Q&A box at the bottom of your screen. Marty, I'll turn things over to you.
Okay, thanks, Kristen. Okay, so today, we're going to do things a little bit differently to a standard webinar, I guess. We've incorporated a number of videos just to try and help give you a little bit more of a live or a regular sort of field day feel to it. So you'll hear from graduate students and different people about tar spot this morning and some of the different efforts we have going on at MSU here. So with that, I'll get started then. Just in case you haven't heard about tar spot before, I mean, this is what you're looking at here on these corn leaves. And it, as the name suggests, it looks like, you know, tar has been flecked onto those corn leaves. And that's the actual reproductive structure of the fungus. And so when we look at a section of one of those tar spot lesions, or those black tar spot structures, this is what we're looking at. So the top side of the leaf here is the top side of the image, and you can see spores being pushed up onto the surface. And within the corn leaf itself, you can see these large sort of circular structures. And this is the tar spot fungus producing spores within the leaf surface, within the leaf structure itself, right? And you can actually see the tar spot structure pushing through to the bottom side of the leaf too. And that's sort of one of the diagnostic keys. You know, when you're looking to identify tar spot, you'll see it on the top side of the leaf. Turn that leaf over and you'll see it protruding through to the bottom side as well. Unfortunately, now that tar spot is here in the US, it's here to stay because it does reside in residue. And we've done some research across the Midwest here demonstrating that it can survive on corn residue over the winter quite easily. And then from that, we'll get initiation of infections of new plants. And from the time a spore lands on the leaf to the reproduction of those tar spot structures and more spores being produced, it's around about 2, maybe 3 weeks, depending on temperatures. So the problem is that means we can have many, many cycles of this disease during the season, and that tends to contribute to that sort of explosive nature of this disease, and it can amplify very quickly. Where did it come from? Well, it's been known in Latin America for a long time now. For about 100 years, it's been described, or over 100 years. And, you know, this is one of the centers of diversity of corn, right? So this is where we get a lot of our different germplasm from. And it's been native to that area, you know, as I said, for over 100 years, or known to be there for over 100 years. And it was only just recently introduced to the US. Back in 2015, we had it turn up in Indiana, Illinois, and then it subsequently spread out of there and has been in Michigan as well. And we'll talk about distribution here in a minute. Before we go much further, I'll invite Emily in just to talk a little bit about identifying tar spot and how you can differentiate it from other diseases or other things on the leaves.
Yeah, so there's a few things that look pretty similar to tar spot when just like scouting in the field. So one thing's like insect frass, so bug poop. And one thing you can do to make sure, or one thing you can do that will happen with insect frass though, is you can just rub it off either like with spit or water. It'll just rub off the leaf. Another thing is also rust. So a later stage of rust is the teleospore stage, and it has black spores, and that'll look pretty similar to those black tar spots on the leaves. But these kind of burst out towards from the epidermis or the leaf surface, and they can be kind of scraped off as well. So both of these things can be scraped off, whereas tar spots will not be. They'll stay on the leaf.
Thanks, Emily. And then the other diagnostic feature too, right, to remember to turn that leaf over, and that tar spot structure should be protruding through the bottom side and sort of have a bit of a raised appearance to it. I'm going to flip over now to a video that Emily put together just about some of her research project. It's a difficult disease to work with because the spores of this fungus look like a lot of other spores. And so she's working on some techniques to help us identify what is going on and to do some research to help us better understand this disease.
Hi, I'm Emily Rogenkamp. I'm a PhD student in the Genetics and Genome Sciences and Molecular Plant Science programs, and I work in Marty Chilver's lab working on tar spot of corn. One of the projects I've been working on is looking to develop a qPCR assay to help detect and quantify tar spot. Tar spot has what's called a latency period, where the pathogen is there, but we don't see the symptoms, so those black stroma are black spots on the leaves. And so this qPCR assay will help in detecting the pathogen when we don't necessarily physically see the symptoms there.
This assay will not only help in diagnostic purposes, but also will help in predicting tar spot infection. Through collaborative efforts, we are using this assay to detect tar spot spores, and we'll measure environmental factors to build a predictive model to know when to apply fungicides for the best disease prevention.
One of the main symptoms we see in the field is those black spots on the tar spot leaf. A secondary symptom that we can also see is what's known as a fisheye, and it's a necrotic lesion around the tar spot. Fisheye lesions are reportedly caused by Monographella matis, but this pathogen has not been observed within the United States. However, fisheye lesions have been observed in Michigan and across the Midwest. Fisheye lesions have previously been thought to cause a higher yield loss, and so I'm working to identify what is causing this symptomology in the United States so we can better prevent it in the field. My other projects are focused on looking at the genetics of the system. Specifically, I am hoping to understand how the pathogen is infecting the corn, but also how the corn is defending against the pathogen tar spot. These studies will help breeders to breed for resistant varieties in the field.
I don't know if anybody noticed then, but not all of those leaves were corn leaves. There's a few leaves in there of a grass species that also has tar spot-like symptoms on it. We believe that's a different species to this tar spot infecting corn, but that's part of the work that Emily's doing, understanding that, trying to figure out why we get these sort of fisheye, that death of leaf material as well, and understanding why that occurs. So in terms of the conditions that really favor disease, from the work done in Latin America, they were able to sort of figure out what weather conditions were likely in that region of the world, Latin America, to contribute to tar spot development. And what they sort of came up with is like these monthly average temperatures, 63 to 72 Fahrenheit. So it appears that it likes it slightly cooler. In Mexico, they don't see it in their summer growing season, more so in their winter growing season or high altitude where things are cooler. High relative humidity, leaf wetness is critical, and it's very important for many different diseases. And the leaf wetness is sort of required for spore germination and infection of the plant. So without that, you might not necessarily get the infection occurring. So the work in Mexico there, they predicted where they think tar spot is going to be an issue in the US. And so far it's lined up with what we're seeing in terms of development. Indiana, Illinois, around Lake Michigan there sort of been some of our hotspot areas.
Marty, we got a quick question for you.
Yeah.
Does tar spot stop reproduction at a certain temperature?
That's a good question, Eric. So I guess we're starting to just question the temperature requirements, right? Those studies were somewhat crude in terms that they were just looking at sort of monthly average temperatures, right? And it's a great study and it's really helpful, but it's just looking at monthly average temperatures. And it probably does, those temperatures probably also do coincide with leaf wetness too. So we're sort of questioning that. It was certainly very warm this season. And I think the more important part of that though, it was quite dry for a lot of the season, and that definitely stopped infection of tar spot and a lot of other diseases too. But that's a good question and something that we're trying to look at. So one of the questions we do get as well is, when can it infect? And here's some volunteer corn, right? That's at the V3 growth stage in a field that had a lot of tar spot in it. And you can see symptoms that have developed on those seedlings. So whenever there's green leaf material out there, we get the right weather conditions, it can infect, right? So I think the earliest we've really observed symptoms in the US here have pretty much been in July, where we really see rapid disease progression in August and September. And part of that is build up of the amount of disease in the field too. If there's just a little bit, 1 in 10,000 plants, we're not going to have much hope of finding that when we get out and scout. But once it starts to amplify and we get, a lot more plants infected, obviously it's going to be easier to find at that point in the season. And just to talk to the explosive nature, this is back in 2018 where we had quite a severe epidemic in Michigan and around Lake Michigan, Wisconsin, northern Indiana, and Illinois. This is a field there. Disease was first observed in July, but it didn't really start exploding until August. And you can see from this aerial shot, it's exploded and moved to the tops of those plants. And that ended up resulting in premature death of that field, premature senescence, and then lodging as well, where things were really severe. And this area of severe disease corresponded with that area of lodging. So another concern. In terms of, what we've seen over time now, unfortunately, it's pretty much across most of our major corn growing counties in Michigan here. We expect this to sort of continue a little bit. It'll be interesting to see how it plays out with Nebraska and their irrigated production. Obviously, some parts of Nebraska are pretty arid, so that'll certainly help them. But how is irrigation going to play into that? 2019, we saw a really big explosion across Iowa too. Very, very low severity, so not much yield loss in 2019. But it continues to move. And same with Michigan up into the Thumb region, right? Last year, it was very hard to find. There was one or two sort of fields that resulted in us lighting up these counties, up in Huron and Tuscola, et cetera. But as I said, 2018, we had a lot of disease pressure, tar spot disease pressure, and lost, it's in the order of 50 bushels in some particular fields. And that was really driven by those frequent moisture events during the season. This year, 2020 has been pretty dry overall too, so it's certainly delayed disease onset and hopefully some of the yield loss we see from tar spot. So I would look at this map sort of as a risk map as well, where we've had greater tar spot for a longer period of time. That's where we have more disease inoculum and we've got greater potential for disease loss. But over time, I'd be very concerned about the Thumb region as well as inoculum builds in that particular region. With that, we'll put in a couple of quick polls. I'm just kind of interested in your experiences out there in 2018 and 2019. So Megan, if you could cue those.
So Marty, while folks are answering their polling question, I've got another one for you. How long, once you have infection, how long until you can actually see those visually?
Yeah, so that's another good point. So I think tar spots quite difficult to scout for just because it's very different to northern leaf blight and grey leaf spot. With those diseases, we typically see fairly large lesions that are pretty easy to identify. Even from a distance, tar spot starts out as really sort of pinpoint lesions on the plant, and they can be pretty easily missed until we start getting, disease developing to a level that's much easier to spot. So that's certainly something to be aware of. And it takes, as I said, about 2 weeks from when the spore lands on the plant till we get one of those tar spot structures themselves. All right, so some data from 2018 then. We've had a few people in this particular poll here, about 77% didn't experience any yield loss in 2018. 12%, 0 to 10, 9%, 11 to 20 bushels, and only 2%, 21 to 30 bushels. That's good. It obviously depends who we're asking, right? I know West Michigan, we had a lot more issues. Megan, let's go to the next poll. I was just curious if that had changed at all in 2019. I'd say in general too, that 2019, as I said, was a pretty low disease year overall. We really didn't see a great deal of grey leaf. We have some grey leaf spot coming in now and some northern leaf blight, but it's been pretty quiet and a little bit of rust too. On the tar spot front, not only in Michigan, but other states, it's been quiet this season. So something pretty similar, I guess. That's what we may expect. One there that's saying 41 plus bushels. Thanks, Megan, for running that. And again, the concern is, if we get a really wet 2021, like rainfall during the season, we may have some pretty significant issues that develop. So that's what we're concerned about, and that's why we want to talk to you about tar spot. This season, this is what we've seen in terms of where tar spot has shown up so far at the county level. So those grey coloured counties are where we've seen tar spot previously and in previous years, and yellow is where we're seeing it this season. So I'd say, Michigan is on par with Wisconsin and other states, in terms of it's there, but not quite at the levels that we saw back in 2018 or quite at the spread yet of 2019. So one of the really, really important components of pretty much managing any disease is plant resistance. So I'll turn it over now to Addie and her student Blake to talk about some of the really important work that they're doing here, screening for resistance. And this is an aerial shot of the trials that we have down at Decatur, where that grower lost probably about 20 bushels last year to tar spot. We've got a lot of fungicide trials in here, and Addie's got a lot of material over this side. You can see it's sort of checker marked with different material in terms of very diverse germplasm. So with that, Addie and Blake, please have at it.
Thanks, Marty. If you could go to the next slide. So as Marty said, we're doing a project to develop genetic resistance to tar spot. And the way we're going about this is we're screening diverse materials to assess whether they are resistant or susceptible. And then the goal is to move that resistance into elite material by doing a series of crossing. While we're doing that, we also are interested in finding the gene regions that are responsible for the resistance, and that will help in future work aiding in selection, but also aiding future variety development. And then we're also working on alternative trait screening methods in order to increase our accuracy and precision and efficiency. And that includes things like drone flights or also image analysis. And our funding for this project I wanted to mention is from USDA, as well as Project Green, Michigan Corn, the Plant Resilience Institute, and a grant to our group at the university. In conjunction with our USDA project, we are working with collaborators in Wisconsin and Indiana, where they're growing a lot of the same material that we are and screening it there as well. So we've got multiple sites working to develop screening for resistance. And we also have that material growing in Minnesota and Iowa and Nebraska. So we'll hopefully see if something happens there. And Marty, if you go to the next slide, I'll pass it over to my graduate student, Blake, who will talk about our progress so far.
Yeah, so last year we planted in Allegan, Michigan. We had about 500 different varieties out there, ranging in maturity and among other traits. We took incidence and severity ratings on that. Like Marty said, disease was a little bit lower last year. We have a little higher disease this year. So we got some data from that. We took some drone surveys with just kind of some lower grade camera work. But this year we upped our varieties up to 800. We have a lot higher ratings. Right here, I kind of have a GIF of kind of the disease pressure throughout the season. Started about, I think, and beginning of August, we took our first rating, and then week 6 was 2 weeks ago or something like that. Disease was a lot higher. Basically, if you're looking at this GIF, you got white plots are basically the so far haven't found anything, and it goes up progressively from there. We have drone data that is a little bit more advanced, some more advanced camera work, and we're going to continue working on crossing with whatever's resistant. If you want to skip to the next slide. So yeah, like I said, basically future work is just going to be processing that image data for just some more rapid assessment on where it is in the field. We're also we're starting to work with some more basic like leaf pictures of just taken on cameras to see if we can get some percentage leaf area of the disease. And then we're going to continue the breeding process.
And that's
Awesome.
it for us, but if you guys have any questions, we're happy to answer them in the chat as we go along or whenever.
Absolutely. And we'll have hopefully if we get done in time, we'll have a little bit of time for chat at the end of this particular TarSpot section. And if not, we'll definitely stay on at the end of the meeting to answer questions. And so I'm going to play a quick clip now too from another graduate student in my lab, Jill Check, who's looking at some agronomic factors that potentially drive disease and epidemiology too.
Jill, you are completing your master's degree with Marty. Tell us a little bit about what you're looking at. So last growing season and this growing season, we're looking at the relationship between planting population, nitrogen fertility, and disease severity of tar spot. So we have trials set up where we have different planting populations from 28,000 to 46,000, and we're going in and individually rating the disease severity in each of those plots. And we have nitrogen fertility rates of 80, 160, and 240 pounds per acre of nitrogen. And we're doing the same, going in and rating the disease severity in those plots.
So what did you see last year? What do you think the takeaways were for fertility?
So fertility, we didn't have conclusive trends from low, medium, high, medium, low, and between the two locations we had them at, one in Allegan County and one in Montcalm County. But for planting population, our preliminary results are showing that lower planting populations are actually having higher disease severity.
Yeah, and that surprised us, right? Because we were expecting, higher plant population, higher density of plants, creating that microclimate, but it wasn't the case, right?
Yes, that wasn't the case. Typically what we expect to see is higher planting populations, more humidity, more fungal reproduction and spread, but
it's the opposite. So what we think might be happening is in those lower plant populations, we've got lower density. We're losing heat quicker from that canopy and potentially having longer dew periods in that canopy because of that.
Jill, can you explain what this contraption is?
yeah, so this is our Burkhardt spore trap. We're using it here out at our Decatur trial site to do some trapping of Phylophora matus spores, the causal organism of tar spot. We're going to try to trap spores to see how the weather around us is causing spore release.
And what do you do once you collect the spores? How do you identify them?
So hopefully we'll be using some molecular tools to be able to quantify the number of spores we're catching in the air.
Yeah, unfortunately this fungus, the spores are pretty similar to other fungi that are out there, so it's going to be hard using a microscope. So using some DNA techniques should be able to help us identify when the tar spot fungus is actually releasing those spores.
And how often do you come out here and check this?
So the way this trap works is it has a rotating drum. So we have it set for every 7 days. It'll do a complete rotation. So I come out here at the same time every Friday to change the tapes and bring the old one back to the lab to cut up into 24-hour segments.
And change out the battery, right?
Yes, and the battery.
So that just gives you a quick sense of what Jill's doing, and that integrates with what Emily's working on too. So at the start there, Emily's project, she's developing, she talked about qPCR assay. It's basically a type of assay that we can extract DNA and then figure out how much of the pathogen is present in samples that we collect. Jill, would you like to just comment? So Jill had done a fertility trial rate where she put down different rates of nitrogen and basically didn't see any differences, but we were surprised at the differences that we saw in the amount of disease in her plant population trial.
Right, so this is our data we collected at our Montcalm location last growing season. So on the left, we have the results of our resistant line that we use, and you can see the clustering between the low population and the high population on our disease severity. And then for our susceptible line that we used, we didn't have the same kind of clustering that we saw on the resistant line, but we do see a trend of higher planting populations with lower disease severity and then lower planting populations with higher disease severity.
Right, and as we indicated too, I mean, this sort of caught us off guard, right? We were thinking along the lines of managing white mold and soybeans, and in that sort of situation, the denser the canopy, the greater the microclimate underneath that for mushroom production and plant infection. But it was just the opposite, right? So those low plant populations had higher levels of disease. So we've been working with Yunsuk Dong from Biosystems Engineering, and he's been doing some work, and I'll switch it over to him now just to talk about what he's been finding.
Yeah, well, thank you, Marty. So I'd like to briefly discuss our leaf wetness sensor monitoring work. So first of all, what is the leaf wetness sensor? The leaf wetness sensor is designed to mimic a real leaf and is basically detecting the leaf wetness. So using this sensor, we can measure the duration of leaf wetness, which can be related to the crop disease potential. So as Dr. Marty Schieber mentioned earlier, the tar spot is common with over 7 hours of leaf wetness combined with the 75% average humidity over a month and the temperature of 60 to 72 degrees Fahrenheit. So we've been looking what environmental condition and irrigation practice and planting population that impacts the duration of leaf wetness over 7 hours. So this year we have installed the leaf wetness sensor in several locations in Michigan. At each location, the leaf wetness sensor are placed at 1/3 height and 2/3 height of corn. So we installed the leaf wetness sensor monitoring unit at Jill's planting population study plot. And we have the sensor unit at high planting population and low planting population. Also, we installed the sensors in several irrigated farms in Coldwater and Sturgis. Each farm, we installed the sensors in the irrigated area and the dry corner to see if or how the irrigation impact on extending the leaf wetness duration. Next slide, please. So I'd like to share some of the leaf wetness sensor data. This is 2/3 height leaf wetness sensor data that we collect from the planting population study plot. The top graph is from the lower planting population and the bottom graph is from the higher planting population. And the y-axis represents the leaf wetness duration in minutes and x-axis is the dates. So I have calculated the continuous leaf wetness duration for each event, which is shown on the top of bar. And I changed the font color to red if it is over 7 hours. The overall, we did not see clear difference between high and low planting population for this 2/3 height leaf wetness sensor data. Next slide, please. This is the leaf wetness sensor data we collect from 1/3 height of corn. Well, this preliminary data shows that in the higher planting population plot, which is on the bottom, exceed the leaf wetness over 7 hours only once, but the lower planting population, which is on the top, exceeds 7 hours of leaf wetness 8 times during this monitoring period. Well, as Dr. Schieber explained in the earlier video, a possible cause for this is that the lower planting population, it's losing the heat quickly from the canopy, which may result in longer dew period. We are going to continuously monitor this leaf wetness sensor on these locations this fall. So once we find some more interesting information, we'll be happy to share it with you this winter. Thank you, Marty.
Thank you, Yunsuk. And just to sort of demonstrate, the potential for irrigation to drive disease, let's talk about an example here. So this is something that Bruce McKellar put together with Jeff Stuck. So obviously looking at an aerial shot here of his pivot and the dry corners, right, marked out with X's there. This is just the hybrid planting map, and you can see this in the next imagery of like plant health. It's pretty obvious, where this band of other hybrid is showing. But what I really wanted to focus on is sort of those dry corners compared to where we're actually irrigating. And remember, again, 2018, we had a lot of moisture in terms of rainfall that season. So there's a lot of moisture and they really didn't need to irrigate too much. But when they did irrigate, they actually saw, a negative impact from that. So when they irrigated, they promoted disease. And we can see that in terms of plant health imagery here. So we've got a lot more sort of browner, darker colors here. Compared to the dry corners of the pivot, right, where we're not irrigating, we don't have as much disease, and that led to differences in yield. So when we're irrigating, we're down to 170, 180 bushels per acre, and in those dry corners, we're at about 220 bushels. So it just sort of really demonstrates the potential for this disease to explode if conditions are right and result in pretty significant yield losses. And here's something that Bruce put together in terms of when we had rainfall events, which are shown in the blue there over the course of the season. And those couple of times in red there where the pivot was run. So, it was only a few times that pivot was run, and that resulted in some pretty dramatic effects there in terms of yield. 2019 is quite a different year, right? This is some photos courtesy of Dan Heasley from 2019. This entire field had headline AMP applied, which is, a fine fungicide, but it sort of demonstrates that fungicides do not eliminate disease. So where we were irrigating, we had a lot more tar spot and saw premature plant senescence because of that, right? But 2019 was quite different. We needed the irrigation last year, even if it drove disease to some extent. Irrigation was still important for achieving yield potential. This was a very light sandy soil, and when they ran the combine across that field, under the pivot, they were still achieving an advantage in yield, even though they had driven disease, right? So it's a really sort of fine balance between irrigating, driving disease, and chasing yield, right? And the dry corners there were lower, right? Because we just didn't have enough moisture in these corners. So we've got multiple anecdotes of irrigation driving disease and some data now from Yunsuk and Jill's work to sort of demonstrate, how that leaf wetness is potentially driving this disease. So that's something that we're going to continue looking at. And I'm going to switch it over now to Lyndon Kelly to talk about irrigation management and the importance of that to irrigate correctly to minimize leaf wetness events.
Thanks, Marty. So Dr. Dong, Steve Miller, and I have been working on some irrigation issues. But one of the things that we talk about, we sure don't want irrigation to increase the chances of disease and result in a yield reduction. We want to see irrigation stay as a positive thing as much as possible. So this work, as Marty and others have found, that we need that 7 hours of wetted leaf time to bring the disease forward. We started looking at how often does our irrigation applications increase that chances of disease, that we actually increase the wetted time. And if you notice, this is one of Dr. Dong's charts from earlier year. The upper chart is the upper third of the canopy and the lower chart is the lower third of the canopy. The green marks, green arrows depict a couple irrigations early in the season. And if you notice, both of those irrigations did not result in 7 hours of wetted time period, 7 hours being demarked by the red hour numbers at the top. But if you look at the third irrigation application, that application increased us to 8.2 hours of wetted time. And if you look later in the season, we had even longer. So one common concept is, well, let's only irrigate when the corn will dry off in less than 7 hours. The challenge here is that most irrigators do not have the equipment and the pumping capacity to be able to keep up with water removal without having to pump 24 hours a day. But if you did have late in the season, corn's using less water, maybe you could only irrigate those hours, say 6 or 7 in the morning till 5 or 6 at night, when the crop would actually dry off before we got into the heavier amounts of moisture in the air and the dew. So that's one concept. Unfortunately, for most of us at peak season, we're not going to be able to do that. So Marty, next slide. So another concept is, okay, let's make sure we're actually irrigating when we need the water. If we could avoid, if we're not going to get a yield increase from that irrigation application, then let's avoid that application, avoid the cost and the use of the water, and avoid getting the crop wet. So that comes back to basic irrigation scheduling. There's a couple new products out. There's this from the National Weather Service. They have an estimation of how much water a crop would use this week. This is actually for, I think it just ends through today. It would say that green grass, which is at this point using about the same amount of water as our corn crop, used 1 inch of water this last week. So we would do our calculation based on 1 inch this week minus rainfall. So many of us are actually positive this week. We got more rainfall than we did, and we wouldn't need to irrigate. But basic irrigation scheduling is one concept there. Next slide, Marty. If you like something simpler, there's a fact sheet online at the irrigation website that Steve Miller and Yunsuk and I have kept up for a number of years. It has a basic chart would tell you that if you had a 75-degree day and you had dent corn, that it needs about 1/10 of an inch or 11/100ths of an inch a day. So that's a quick way of looking at that same type of concept. And those are the calls that I've been getting the last week here. If I put it on an inch last week, how long will it last? Well, if it's a 1/10 of an inch a day, 10 days, that it would be fed by that 1-inch application. Next slide, Marty. So the other concept there that can work in our favor is to make sure we're putting enough water on now so that we don't have to do multiple applications. If we think about a typical August here in Michigan, our corn crop would use about 6.3 inches of water. So that's the last 5 years of water use in August across southern Michigan from Lansing down. In a typical year, you'd get about half of that, about 3 inches of rainfall. And some of us got shorter this year, some of us did really good. But in a typical year, you need to put about 3 inches of irrigation on to make that need. If you do that in 3 1-inch applications, you got the crop wet 3 times. If you did that in 1/4-inch applications, the other extreme, your 12 wetting events. The advantage also is that every time we wet the crop, we wet the canopy and the surface of the soil, and that water never gets to the crop to be able to use for evapotranspiration. So we actually get, the larger applications actually get more water into the ground for our crop. And Marty, last slide. Some of us that have center pivot irrigation are going to think about applying a fungicide through the system. I don't have enough time to talk about that whole process. There's a excellent presentation online at the irrigation websites at both MSU Extension and Ag Engineering and Purdue Ag Engineering that talk about that process and meeting the requirements of the fungicide label. When you're doing the application. The big issue here is many of our pivots do not apply small enough uniform applications of water. Some of the products, you're not supposed to be using the end gun at the same time. So these are all challenges are there. Other things, you need to have the safety check valve. That's a requirement in all the states. Need to have a positive displacement pump. And we need to have safety systems to make sure that if one component of the irrigation system shuts down, all of the components shut down. So go look for more information at that website and we can take questions later.
Thanks, Lyndon. And we do have some key mitigation trials out with the grower or demonstration type trials this season. So I'll be really curious to see how they pan out in terms of tar spot management. I know we've got good disease pressure at that location. So with that, I'm going to turn it over to a video that Bruce and I put together. Bruce is pretty hard to find online, so I had to have my 5-year-old put a picture together, a headshot. So let's go to that then.
I am down in Southwest Michigan in Decatur with Marty Chilvers and Bruce McKellar looking at tar spot. Marty, can you tell us a little bit about the trials that you've got going on in this field?
Hi, Christian. Yeah, absolutely. So we're down at Decatur. We're doing all of our, or most of our trial work for tar spot of corn. So in here we've got a number of different trials over about a 10-acre field here. We've got fungicide comparisons, so different fungicides, seeing how they perform. Fungicide timing, trying to like really work on optimizing that fungicide timing, because that seems to be even more important potentially than the product you pick. Then we've got epidemiology type projects where we're trying to figure out when spores are being released. Agronomic practices too, such as the amount of nitrogen, does that drive tar spot or not? And then plant density too, plant population, does that have any impact on how tar spot develops? And then we've got variety screening too. We've got both commercial hybrids here that we're doing screening of to help companies figure out which varieties have susceptibility or resistance. And then we've also got like a lot of material, a diversity panel that Addie Thompson's group is looking at to look for new resistance sources to tar spot. So a lot of different things going on in this particular field.
So first let's talk about some of the pressure that you're seeing. How would you rate where you're standing right now, or the leaves that you've got?
Yeah, Yeah, so here we're starting to see pretty good pressure now, right? So you can see tar spot really starting to develop on these leaves. In some situations like here, it's starting to prematurely kill that leaf, I guess, starting to shut the plant down a little bit. And pressure is running from the bottom of the plants. This was corn last year. We had tar spot in here last year, probably affecting yield by around about 20 bushels was the farmer's estimate. So it started from the bottom, from that residue, that trash that's on the floor there, spread up onto the leaves and has moved its way up to the top of the plant. So I can see lesions all the way up on the flag leaves up here now. And sometimes when things get bad, we can even see lesions develop on the tassels. So those black spots moving up. So in terms of rating, we do it a couple of different ways. We'll go through and look at individual leaves perhaps. Typically we'll look at like an ear leaf and then rate that for how much tar spot is in there. We'll give it a percentage severity rating. There's that style of rating we do, and we also do like a plot-wide estimate, walking the plot and getting a sense of how much disease is in here. It depends on the trial specifics, but that's a couple of different ways. We also have drones flying weekly too to capture overhead imagery to get a sense of how it looks from above.
Talk a little bit about the management options for tar spot.
Sure. Fungicides is certainly one of the main things we think about. When we're ordering seed though, that's a great time to think about hybrid susceptibility or resistance to this disease. There's no hybrid that's completely immune to tar spot, but there are differences in susceptibility. So definitely talk to your seed dealer about that. But when we're talking about fungicide treatments, there's a number of different products that look pretty good. I mean, most products seem to have some reasonable efficacy for disease. But I think one of the more important pieces of that is timing. So there's temptation, I guess, to potentially put a fungicide on, a V6, V8 when we're putting, herbicides on, but it's really too early in general for tar spot. So I think typically our most effective window is going to be somewhere from reproductive, the start of reproductive, so R1, silking, tassel, through maybe to R4, which is doe, which is really quite late, and we don't often consider that, right? I mean, typically most guys put applications on at R1 and then call it good, but we had an instance last year where that R4 timing seemed to really pay off because we hit it just when disease was developing. So essentially what we're trying to do out here in these trials is get a sense of what fungicide products are working and which products are working better. But I think even more importantly than that, like what timing, when should we put those products on? So again, we're going through and doing a lot of ratings to capture how much disease is there and how those products stand up and how they stand up over time and then how things look at the end of the season.
So Marty, is there a reason that the later seasons are showing higher tar spot pressure? I mean, is there anything environmentally that's going on that?
Yeah, I think there's a couple of different things. So first and foremost, I think at the start of the season, there may be a little bit of tar spot there, right? But it's such a low amount. And I should just back up a little bit. When you put a fungicide on, you do not eliminate disease, right? You knock it down a little bit and you gain protection for a couple of weeks, right? We've got such a long growing season and we don't have the margins in field crops to spray them multiple times. So that's part of the real trick, isn't it? So tar spot seems to get going. We tend to detect it in July, but it doesn't really seem to ramp up until August or potentially even September, right? So it seems like some of those later timings of fungicides might be more important than going really early and then your fungicide has sort of run out of gas and now the disease is starting to develop. And to sort of get more to your point there, certainly in August we have longer dew periods at night, right? We tend to hold more moisture in the canopy, and that's one of the really key drivers to this disease, that leaf wetness period that really helps to drive disease and another reason why we see it start to really amplify late in the season, August, September.
Yeah. Shorter day periods and longer nights.
Yeah.
And more fogs in the morning, right?
That's true. Yep. Absolutely.
So all those things kind of contribute.
Yeah. All right. So just a couple of things to sort of wrap up here. So last season, we looked at the timing of fungicide application. And I think really at that point in time, we were trying to really look at vegetative compared to, R1, perhaps R2 timings. And so this is data from last year from across multiple locations in other states. And I've lost some of my captions here, so I'll just tell you what they are. But the blue bars here are the amount of disease over the course of the whole season. So we do multiple ratings and then add all that up essentially to figure out how much disease occurred over the entire season, because that entire season is going to be what determines yield, right, in terms of photosynthetic loss. So our non-treated check there had the most amount of disease over the entire season. This next bar here was at V6, and it's no different to our non-treated check. Then we had one that was sort of model determined. So we've got this disease model. It triggered a little bit too early, so it was somewhere around the V6, V8. We look at V8 here. We're starting to see a reduction, no statistical difference. It's not until we get into V10, very late vegetative stages, that we start to see a difference compared to the untreated check. And then really these last three timings, that was at R1 and one of these was at R2. So really we were just seeing the best timings generally being within that reproductive stage that we were talking about. And so this year, that's what we're really focused on and diving into that in more detail. So how does R1 compare to R3 or even R4 this particular year and across many locations, and how is that going to play out in terms of what we recommend in terms of spray timings? We do have at this website, the cropprotectionnetwork.org, we have fungicide efficacy charts for corn, soybean, and wheat. So you can go in here, download a PDF, and we've got a number of different fungicides and how they rank for the control of different diseases. So we've got that up there for TarSpot, but based on last year's data, we weren't too keen in really separating things out because it was only a small amount of data and there's a little bit of conflicting information. I'm very sure by the end of this season, we'll have a much better sense of which fungicides were outperforming others and a better, clearer ranking of those different products. So look out there for that, more information. And just sort of a final note, don't forget about other diseases, right? We've really focused on TarSpot because it's new and we've got to figure out how to manage it. But, don't forget about northern leaf blight in this example. So on the left-hand side, a susceptible hybrid, on the right-hand side, one that's far more resistant. So don't forget about those other diseases. And just sort of the take-home points for TarSpot management then, weather is going to be critical in terms of driving disease. And if you're an irrigator, that's one of the things you might be able to sort of minimize the leaf wetness periods when you are irrigating, something to keep in mind. Perhaps in terms of what we can do proactively, hybrid selection is going to be the most important component. And I know over the last couple of years, we've done a great job, the companies have done a great job in screening through and trying to remove some of the very susceptible hybrids. But we don't have anything that's immune to disease, right? We're still learning as things develop. So look for that. And then Scout, making sure we're on top of that. We sort of talked to you about how to look for disease. And again, for well-timed fungicide application, it's going to be somewhere between R1, maybe through to R4. Crop rotation and residue management, we haven't talked about that much here, but that appears to play a pretty minimal role in terms of TarSpot management. I'm not saying that it's not important, but we have seen in corn, soy rotations that we can still get tremendous amounts of disease. We had plenty of people reporting in 2018, fields that hadn't been in corn for many years, planted to corn and getting smoked with TarSpot. So unfortunately, these TarSpot spores from that fungus can move many, many miles, right? And so even if you're, doing a great job in terms of residue management and crop rotation, it doesn't mean you're going to escape the disease. So they may play a role, but I think it's going to be fairly limited. And then this time of the season too, we really want to be scouting for lodging potential, just keeping an eye on those fields. Now that we've got a lot more moisture, September and dew periods, longer dew periods, disease can amplify pretty quickly and that can result in weak stalks and potential for lodging. So scout for that and try and harvest those fields that are going to be at greatest risk to lodging. And with that, I'll just thank again the different funding agencies that made this work possible, and we'll open up for questions for a couple of minutes.
All right, thanks, Marty. So again, please go ahead and take the opportunity to click on that Q&A button on the bottom, type in your questions. While you're doing that, I've got a couple in the queue here. So Marty, how likely are we to see a positive return on investment with the fungicide application when corn is dented? So a lot of the fields that we're seeing right now are at dent. So basically the question is, how late can you apply that and really see some yield benefit from it?
Yeah, I think we're, for the most part, I think we're really going to be beyond the window of a positive return on investment. So when we hit the beginning of dent there, we still do have, you know, somewhere in the order of 40% of dry weight to set in that kernel yet. But as soon as we, you know, as soon as we get the half milk line, I think there's only like 10% or something to go. So depending on exactly where we are in that period, I think the return on investments going to be pretty minimal, the chance of that return on investment. You know, last year we had this instance where a field was sprayed August 20th, I believe. So Jason Roth had set this trial up. It was really a very interesting trial. And so what really sort of came together there is that they did spray it earlier at R1 and there was no disease present, right? And so the fungicide's probably worn off. And then they started to see disease starting to develop around August 20th. And then they went in and sprayed. So this is probably really at the R4 growth stage. We've got some kernels just starting to dent. And they made that application August 20th. And from that trial data, I'll summarize it quickly. They essentially saw a 20 bushel protection from that, right? This year, we're probably a week ahead of where we were last year in terms of heat accumulation too. So I just think the chance of return on investment are pretty minimal. We've had some conversations with folks out in Wisconsin this year as well about trying that in a field or two. I think if you're going to do that, you know, I would be pretty cautious. And if you're going to do it, I mean, make sure you leave check strips so you know what any potential benefit you are seeing. I think that's really critical. And I think anytime you spray a fungicide, if you can, it's a great idea to try and leave some check strips, even if you're spraying in a more sort of normal window, so you get a better sense of how that how those products are performing for you.
So I'll throw another question at you. So we've seen the maps that have how many counties and which counties where tar spot has been identified. Do you have any sort of sense as to how pervasive the tar spot is in these different counties? I mean, you know, for example, in Berry County, was that just one field or is it pretty widespread? Do we have a sense of that?
We have a sense of it for sure. I mean, I think, you know, where we've seen more disease over the seasons, then we've got, you know, a lot more of those fields are infected. So like the west side of the state, I'd say there was disease in every field last year or even the year before. Actually in Allegan County, it was just wherever we looked, we found it. I'd say that's probably even true here in Ingham County. My technician Adam does a lot of very intensive ratings. And he first saw disease in 2018, but it was right at the end of the season, October. He's picking up one or two spots here or there. Last year we had more of it, nothing certainly impacting yield, but it was very easy to find. I mean, this season, I think we found it a little bit earlier yet again. So what is happening with time is that the amount of inoculum is increasing, even if it's not impacting yield because of the dry conditions. So it's just going to set us up for the potential for greater yield loss if we do get a wet season like 2018. So this is very true in the thumb. Yeah, some of these counties are probably positive, right? But it's all a matter of people actually seeing it and then actually reporting it back to me as well. But it's a pretty good estimate of what's sort of happening here, and it really tracks well with what we're seeing in other states too.
So Bruce has a few comments in here. I'll just touch on a couple of them here. He's mentioning that incidents that he's seeing is ramping up even since Labor Day, and a lot of what he's seeing is east of M66 very late in the season, surprisingly widespread in terms of numbers of fields infected.
Yeah, I mean, yeah, that's not a great surprise, and I think that's what we really saw in Iowa last year as well, right? So, you know, before before 2019, I don't think we had any Iowa counties confirmed, but last year, some folk did a lot of driving and found it fairly easily at very low levels, but it was present. What was your question there, Eric? Sorry.
So I'm just going to comment, just looking at the forecast here, we've got, a decent amount of rain that we've just got, with maybe one more chance coming up in the near term, and then cooler temperatures. Just what is sort of your prediction as to what's going to happen in the next couple of weeks until we get to black layer?
Yeah, I mean, tar spot's going to continue developing, right? Wherever there's green leaf material, that fungus is going to infect and continue to move on those plants. And if you've got like a reasonable number of plants infected in the field, that can explode very quickly and potentially result in those stalk quality issues. So really be on the lookout for that. If you've got very, very low incidence of disease, very few plants actually infected, then yeah, I mean, it will continue to increase, but it shouldn't really give you too many issues. So it really just sort of depends on how much pressure is out there now as to how much that has that potential to explode in those particular fields.
All right, I'm not seeing any more questions in the chat or the Q&A.
Okay, should we switch over to our other section then? We can always touch back on this at the end.
Yeah, why don't we do that, Marty? Let's switch gears away from tar spot for a little bit, but thank you, Marty, and everyone who has been working on the management of tar spot. That was a lot of really great information that was covered there. Now, though, we're going to hear from Dr. Manny Singh about another important management decision as we collect data from the 2020 season and select the best hybrids for 2021. So Manny, take it away.
All right, well, thanks, Kristen. It was a very nice first section, Marty. A lot of good information. So I will be talking about some of the work that we have started looking at the optimal maturity selection, which is one of the most important component in hybrid selection in corn, right? And it can become especially more important as we deal with these new issues related to tar spot and even some of the more variability in weather that we have been noticing here. If we review our 2020 planting season compared to the years before, we did a pretty good job in terms of planting, right? Way ahead of our 5-year average. But we know how 2019 went, and even 2016 and 2017, we had some delays in our planting. So essentially what we are looking is that this extreme weather events, essentially more rainfall during the spring, is causing some of these planting delays, right? So we need to be thinking about that. Do we need to change our management based on when we are able to plant, right? And how to deal with that variable planting window. Especially in this research, we are looking at that optimal hybrid selection that we can use to best utilize the growing season that we have. And we are dealing with a relatively short growing season as we are so far north in the corn production area, right? And again, use of this maturity is gonna change or switch when the crop is going to be reaching that maturity time, right? Early September versus end of September. And that might have some consequences in terms of the weather it's observing, what Marty's group was talking about. So if we try to like put our planting time into these 3 buckets of early, mid, and late season planting time, we'll notice that we're dealing with different field conditions, right? Cool and wet soils early, maybe optimal conditions mid-season, lack of moisture in the late season. And we have to deal with that. But more importantly, we are looking at a different length of growing season. And again, when the crop is going to be reaching maturity. So based on that, we need to be thinking about that, okay, how we can use that longer growing season that we have when we are able to plant early, or how do we deal with that short growing season that comes along with the late planting, right? So we need to be thinking about this sort of a portfolio approach of maturity selection to optimize our growing season based on our planting time. And since corn growth and development is driven pretty much by heat units, right? What we call growing degree day accumulation, we can look at some of these growing degree day accumulation maps of our growing season from May until September and see how they are shifting with the change in some of the weather patterns. On these figures, you can see some of these colors are turning a little bit more darker as we are moving towards these new decades, indicating that we are accumulating a little bit of more heat as the growing season is progressing, right? So how we can utilize that extra heat that we might have. Also, we are seeing a little bit of a delay in that first frost event that we do observe. So essentially, how we can match our hybrid selection with this growing degree days, right? And thinking about that, should we be using the growing degree day ratings that all the seed companies do provide on their seed label compared to the use of the relative maturity days, right? And those, I mean, days are misnomer there because it's not the actual calendar days. It's just a relative rating compared to another hybrid within the same seed company. So we got to be thinking about this model of using growing degree days to black layer essentially in hybrid selection. And what that model does, it also helps us when we are dealing with late planting or replanting situations like some growers dealt with even this year. With the late planting, we have this phenomena, what's called growing degree day compression. Essentially what that means is a given hybrid when planted late in the growing season will take less number of growing degree days or heat units to reach black layer maturity compared to if it was planted early in the growing season. Mostly for black layer, but a little bit of that happens even until that silking time. Some of the data from Indiana and Ohio that Bob Nielsen's group did a while ago, actually, early 2000s, showed that this number of around 6.8 growing degree day decline for every day delay of planting. So if the planting were to occur May 30th compared to May 1st, we are looking at about 200 less growing degree days for the same hybrid to mature, meaning that we can use that same medium to long maturity hybrid even in the later planting window. But we need Michigan data, right, to see if that number is about the same. Plus, we need to look at the new hybrids that are available now. So this year, we started this pilot study to look at this interaction of planting time and hybrid maturity. We used multiple planting times and picked 4 different hybrid maturities, all the way from 21 to 2600 growing degree days. So sort of low and high end of what we use around here in the Lansing area. We are collecting a lot of data this year, looking at the phenology, right? When anthesis occurs, when silking occurs, and eventually when the black layer is occurring. So a lot of that data is ongoing. We'll also be looking at some dry down curves and looking at yield and final moisture. And we plan to expand this research next year based on the data we see this year. Quickly, some of the preliminary data that we are seeing. So here I got time to silking on y-axis in terms of, again, growing degree days. And on x-axis, I have my 3 planting times, early May all the way to early June. And the 4 different lines represent the 4 different hybrid maturities that we use, going from the earliest one at the bottom to the late season or the full season at the top. If we were to observe a growing degree day compression, we would see a negative slope on most of these lines with the delay in planting. We did not see that. As you can see, most of these lines are flat. So at least we are not seeing any compression or less time with the delay in planting, at least for silking time. We are starting to get to black layer in some of these hybrids already. And you can see at least for our early maturing hybrid, that 85 CRM or 2100 growing degree days, we saw some compression happening, about 5 less growing degree days per day. So that number is a bit smaller than what Bob Nielsen was representing in Indiana. And again, this is just based on our second planting. So we need to be looking at this data when the whole picture comes through. But you can also see here, like by changing this planting, by changing our maturity selection, we are moving the window of when these crops are going through that phenological development and how close they are to maturity in early versus mid or late September. And that might have some implications in terms of managing a disease like tar spot. One of our goals with this project is to create more data to update this tool called Useful to Usable, which is a very handy tool in terms of selecting maturities for a given farm, right? Basically by entering location, growing degree day, start time, which is the planting time. And then I would recommend again using the actual silking growing degree days and black layer growing degree days instead of using that maturity rating. But this tool does not account for that need of fewer growing degree days if that is happening with the delayed planting. So our goal is to update this tool by collecting again more data from Michigan. But again, I will highly recommend using this tool in the short term in deciding what maturities to be used on a given farm. We are also collecting a lot of data on kernel moisture dry down because, right, it's not only about the yield. We are also spending money on drying that grain. So we want to know, once we get to the black layer, how the dry down will proceed. And here's a picture of data from Iowa where they are showing anywhere from 0.4 to 0.7% decline per day. But we need, again, more data on what happens under Michigan conditions. Look at the effect of environment, temperature, relative humidity, as well as some of this agronomic management in terms of our planting times and hybrid maturities. So we'll have more of this data to share. This year, we also got our hands on this sort of a neat in-field kernel moisture sensor. I can call it on-the-cob estimation of corn moisture. We are in the process of validating this and see how close is this compared to the actual oven dry moisture content. The company has a lot of data on it, but we are trying to make sure that it does what it is supposed to do. And this will again make things a lot easier that we don't need to harvest and thresh kernels to get to a moisture. We can do it just on the cob. So again, we'll be updating this and the final yield data on our website. We also run the Michigan Corn Performance Trials where we use multiple maturities at each of these locations, right? So we have an online database where you can go and look at even a simple maturity comparison, even if the hybrid you are growing is not part of the trial. So we'll be providing more information on this in the winter extension meetings. And with that, I'd like to thank everybody and see if there are any questions.
We don't have any questions yet in the Q&A in the chat box, so everyone, again, feel free to put those in. But all of the presenters, starting with our first one all the way through to the end, if something occurs to you, feel free to add those in as it comes to you, because we'll pick those up at the end during our final Q&A as well. Manny, I'm not seeing anything right now, so thanks a lot.
Sure.
Looks like there's one question that came in. It said, can you explain the Michigan map bottom left of the last slide? If you can bring up that last slide, they have a question about the Michigan map on the left side.
Oh, okay. So this map is pretty much, let's see. Because yeah, this map represents the locations that we run as part of our corn performance trials. So we divide the state into these different zones based on the heat accumulation, right? More in the south and less as we go further north. And we generally pick 3 locations within each of these zones to represent multiple locations, right? And the different symbols represent our grain or silage trials, or locations where we have both grain and silage. So again, this goes back to the idea that if we're testing the performance of a given hybrid, we want that to be a multi-location, multi-year even, which is becoming more of a challenge because of the high turnover. So we at least try to generate data from multiple locations to see the hybrid performance. And we have, again, within each of these location or zone, we have multiple maturities that we evaluate. So again, if you go to our website, you can find a a database where you can search for a specific hybrid at this specific location or multiple locations within a given zone.
And Manny, are those numbers 2020 or are those historical?
So these are 2019 numbers. In 2020, we were unable to plant a few locations because of the COVID-19 restrictions that we were put under. But yeah, these are more like historical locations, yes.
And through what date?
Date, you mean year? So these locations again are until 2019. And this year again, we have at least 2 locations within each zone, but not all 3 of them.
I think maybe they were asking how late into the season you're accumulating those numbers.
Oh, okay. So you're talking about the growing degree days. So the numbers that are represented here, and I was even showing the maps before, they go from May 1 through September 30th. So we are accounting for those 5 months for our growing season. And again, these numbers on this figure are historical numbers. They are the recent climatological normal, which is 1980 to 2010. I believe our climatologist, Jeff Anderson, is about to update those numbers when he has the full 2020 season here. So those will get updated, and those numbers, I expect them to go a little bit higher based on the earlier charts that I showed.
All right, while this video is queuing up here, just appreciate all the questions that are coming into the Q&A. So that's what keeps this lively and active and pertinent for you guys. So keep putting those in. We've got a couple of questions that are focused on tar spot. And so again, thanks for those. We'll hit those when we come back to our general Q&A at the end. So any questions that you have throughout those, they'll end up being anonymous. So don't worry about, other folks, knowing who asked the question. So just keep putting those into the Q&A and we will come back to those.
Okay, we are going to move on to the second half of our program today. Another topic of concern for numerous corn growers this year has been uneven corn growth and development within fields, which can be due to a variety of possible causes. We have MSU Extension specialists and educators who are going to address some common causes for uneven development, what impacts this may have, and what you could do about it. So first up will be Bruce McKellar, Field Crops IPM educator based in Southwest Michigan, who will describe what we're seeing in the fields and what some of the agronomic causes are for it. Next, we'll hear from Aaron Burns, the Extension weed specialist, who will discuss impacts of uneven corn development on weed control. And finally, Chris Defonso, the Field Crops entomologist, will explain how uneven crop development in cornfields can make efforts to control pests such as western bean cutworm and other ear borers particularly challenging. So take it away, Bruce.
Thanks, Kristen. I'd like to talk to you today about a problem that we've seen across many fields in Michigan this summer, which is uneven corn growth and development. In some fields, it's pretty easy to assign what might be causing challenges or differentials in growth. In others, where the soil is consistent and there's not a lot of slope in the field, it can be a little bit harder to understand. This is a situation that we saw quite a bit in the southwest part of the state, where soils at the bottom of the hollows in the fields that have a little bit more heavier soil texture, a little higher organic matter, perhaps a little bit better CEC, they had better growth and more consistent color. This is more typical of what we see when we see water issues in the field, where you have a drown out area, and then also you have an adjacent area where the water ran off and we had less growth. You can tell that's next to a waterway that was left in the field to prevent erosion. Patternation is also important to help understand what's going on in fields. If you have differences in plant height that are associated with the direction of travel or the direction of the rows, then perhaps we should be looking at fertilizer applications and herbicide applications for inconsistencies. But in other areas in fields, we may have to dig a little bit deeper to find answers if it doesn't hold true with any of those or topography. One thing is certain this year, we definitely had cooler than normal temperatures through pretty much the end of May. So early planted corn was likely to have experienced a little bit slow growth. And when plants slow down in the field, it's important to understand that, especially at the time of pollination, there can be implications for yield. Those silks that are not completely fertilized will never develop kernels, and that can lead to yield reductions. Well, we've seen an increase in wet conditions over winter and going into spring, and it's been hard for guys to be able to find the opportunity to plant at periods of time when we can get good stands. And due to this, I think people have started to look at planters that have the capability, at least, of going in and planting no-till, where they can utilize the time, the good weather window, for getting seed in the ground instead of just doing tillage and hoping for better weather to come subsequently. But even with an excellent seed bed, we can still find challenges with emergence in terms of being even. Cold temperatures, especially as we plant earlier in the season, can cause potential challenges with chilling injury by the first water that's imbibed in the seed. And often we think about uneven stands as being a problem associated with either the planter or the conditions at planting time. Planting too wet, we know that can cause several issues, and we looked at that a little bit at a program we did at Ag Innovation Day in 2019 on MSU's campus. Well, singulation is an important aspect of the planter, and we want to make sure our units are functioning properly so that we can reduce the variability in between plants in terms of spacing in our operation of our planter. It's also important to keep a particular attention to the closing system so that we don't have open seed slots and potential for challenges there because of poor seed to soil contact. Especially on no-till systems, we have the ability to change up some of the closing wheel configurations to improve seed soil contact, especially under wet conditions. Planting too wet can also cause issues with sidewall compaction, which can create this hatchet-shaped root pattern that allows us to have plants that just are variably effective and have variable growth over the course of the season, especially under dry conditions. Also, what we do with soil residue, if it gets pinned next to the seed and the seed channel, can cause situations of uneven emergence and growth. So far, we've been focusing on too wet conditions at planting time. It's also important to think about what happens to plants when we're planting under extremely dry conditions, especially with those fields that have been tilled. We have potential for clods to interrupt seed to soil contact, and if that happens, we can have variable time of emergence based on moisture availability to swell the seed and begin the germination process. Also, we can get into situations in dry conditions where you get rootless corn or floppy corn, and that can occur when there's not enough adequate moisture to allow the plants to establish a good root base, and it can lead to uneven and certainly unthrifty corn seedlings. Compaction is a whole nother story. Certainly we know that we have challenges with compaction when we have wet falls, and we have limited time to be able to work on fixing some of those ruts. But it's also important to keep in mind that when we're pushing the envelope on planting, that if we do tillage when the conditions are too wet, we can create compaction in the spring too. Compaction has long legs, if you will, throughout more than perhaps one growing season. So it's important to understand how compact fields are and look at what that may mean for productivity over time. We can also have challenges from winter annual weeds and cover crops where they're not consistent across the field. that can cause differences in soil moisture at planting time, and perhaps shading and cause soil temperature differences. We don't often think about rainfall as being, vastly different from one part of the field to the next, but certainly with heavy localized rain events, especially in low wind conditions, we can see that. And heavy rainfall can have season-long impacts, even though the duration that the corn is underwater or you have flooding conditions can be pretty short. And that's especially true where we have nitrogen applied, where the nitrogen can leach out of the root zone or potentially denitrify. Of course, wildlife creates a whole bunch of different set of challenges. We can have problems with sandhill crane pulling the plants out in uneven areas in the field, and also issues with deer. And deer, especially in fields that I've walked in Southwest Michigan, can start feeding on corn leaves in the vegetative stage, and that sets the stage up for uneven plant height in large portions of fields, especially near woodlots. And finally, if we're looking at variability in plant color and plant growth, a lot of times fertility can be a significant issue there, as well as potentially herbicide injury. As we can see that areas that are very adjacent to each other in the field with differences in coloration early can have significant differences in plant growth and development and yield potential. To talk a little bit more about some of the factors that impact this, we're going to switch to Aaron Burns, our weed control specialist for corn, to talk a little bit more about how herbicides can impact this situation.
Thanks for that question and observation, Bruce, on the impact of uneven stands and weed control. My name is Aaron Burns, and we are going to discuss that topic in these slides. We know that uneven corn stands can impact weed control mostly in two categories. And the first is that uneven stands makes weed control difficult. This is a drone image from a field that has an uneven stand near St. John's, Michigan, in which the lighter areas, which I am circling now, are a dense infestation of weeds. So what is the impact of this on that uneven stand? And the first thing is we know that uneven stands are not as competitive with weeds, as you can see in this picture, in which we have a poor stand establishment. We have a heavy, dense infestation of weeds. And that is because weeds that emerge prior to the crop or shortly after the crop are the most competitive. In a study that was evaluating weed control height across a number of states within the Midwest, found that if we applied a post-emergence herbicide to 4-inch weeds, we had about a 3% yield loss. 12-inch weeds, 21% yield loss. And then if we waited till 14-inch weeds, we had about 30.5% yield loss. So the taller those weeds are, the more yield loss we might observe. Also, large weeds are just difficult to control. On the left is a picture of 4-inch Palmer amaranth, fairly small. We have herbicide options to control this weed at this height, as opposed to the right side of the picture, which is 12-inch Palmer amaranth. You can hardly see any of the soil surface. These are going to be much more difficult to control with our herbicide options. The second implication of stand variability and weed control is herbicide injury. And we know that uneven corn emergence and growth stage at herbicide application can play a large role in how these plants recover from that herbicide and also continue to grow throughout the season. This is a picture from a field in southeastern Michigan in which this early post-herbicide application was made all the way from emerging plants, so VE to V4 plants, and I have those plants circled in the orange circles on your screen. And we have many herbicides that have cutoffs, both minimum and maximum corn height and stage that they can be applied at safely. So if you do have an uneven stand, checking those herbicide labels and also the MSU weed control guide to ensure you're still in the correct window for that herbicide. Also, environmental conditions play a role. So cool, wet soils can lead to corn injury from pre-emergence herbicides. Plants are able to take up that herbicide at a rapid rate, but aren't able to metabolize it quickly. So then we see this type of injury. And there are two sites of actions of herbicides that often cause this type of injury. The first are group 15, so long-chain fatty acid inhibitors. These have active ingredients of metolachlor, acetochlor, and dimethylamine P, which in the parentheses I have the trade names listed for these herbicides that contain these active ingredients, and you can see there's quite a few, and they cause this twisting of the leaves that you see on that photo. The second group is group 2s, which are ALS inhibitors, so thiocarbazone, rimsulfuron, flumetzolam. Once again, in the parentheses are the trade names of those herbicides, and the injury is often this chlorotic yellow tissue, purple mid-veins, and then if you dug up some of these plants, you may see some stunted roots. Injury increases with cool, wet weather, shallow planted corn, and also the number of other agronomic implications that Manny spoke about earlier, and also with lighter, lower organic matter soils. But on the good side, if the growing conditions are good following the weeks after this herbicide injury, we often see these plants recover and have little or no impact on yield. But this is not always the case. So you might drive past a field throughout the summer and observe that sometimes these plants don't recover, and we do see some implications on growth and emergence throughout the season. This is a picture of a field from southeastern Michigan, which is on your left-hand side, is a little bit lighter. Lower organic matter soils, we do see some implications with corn growth, as opposed to on the right, some darker soils. And we see that the plants are growing a little bit better, and this was taken in June. And then the same field was followed to August, in which we have some implications now on corn ear development. So some variable corn ears on your left-hand side with those lower, lighter textured soils, as opposed to the right side, which had those more darker soils that were growing a little bit better in the beginning of the season. There are a number of implications to these and reasonings behind fields that don't recover. The first are environmental conditions. So if the weather stays cool and wet for an extended period of time after that herbicide application, we can see some extended implications on injury. And the second, which is very important, is soil texture and organic matter. So those lighter, lower organic matter soils, we need to make sure that we're applying the correct herbicide rate for the soil conditions. And then interactions with soil fertility and nutrient management. So when corn plants are stressed, they need the correct nutrients to outgrow that stress. And that includes stress from herbicide injury. And then oftentimes, if there are not the correct nutrients in that field, we do see some symptomology that can be very similar to herbicide injury. So also investigating what soil nutrient profile is also important. Some recommendations for avoiding these injury and also implications of weed control and uneven stands is use the recommended herbicide rate to avoid corn injury. So knowing your soil texture and organic matter is very important. If the weather is cold and wet, we can choose pre-emergence herbicides that contain a safener. These safeners help those corn plants metabolize that herbicide and continue growing at a rapid rate. And examples of herbicides that contain safeners are Dual 2 Magnum, Cinch, Harness, Break Free, and Surpass, and a number of others. And finally, we have a lot of other resources about the weed science team at MSU, and you can find these on our website, just canr.msu.edu/weeds for all weed science information. Thanks for listening.
Thanks, Erin. And now we're going to hear from Chris DeFonso, MSU's field crops entomologist, to learn a little bit about how insects can impact corn stand evenness. Chris?
Well, thanks, Bruce. So stand variability and insects. Some insects cause stand variability in corn, and one of those is Asiatic garden beetle. When Asiatic garden beetle feeding is really bad, you actually can have plants that are dying in the stand, as you see in this picture from southeast Michigan, but this isn't very common. What's more common is moderate feeding on the roots, as you see in this picture right here, where there's root pruning, and the plant survives, but it's stunted and shorter than the plants around it. And this is a great picture that demonstrates that, where these two plants have been severely fed on, and they are either going to produce no ear, and if they do produce an ear, it's going to look like this. it's probably going to be out of sync with the rest of the field and therefore pollinate late, and that tip doesn't fill. So the field may look fine from the road. All the plants may appear to be there and they're tall by July and August in the season, but what you're not seeing is this hidden yield damage where many of the tips simply don't fill because of that stand variability caused by grub feeding. An insect that profits from stand variability is western bean cutworm. It doesn't cause it, but it would enjoy having a variable stand. Western bean cutworm is now our primary ear-feeding caterpillar in Michigan. Most of the BTs don't kill it, so we get this tip feeding into the side as well, and then the association with mycotoxins. The moths are pretty picky initially about where they prefer to lay eggs. They like to lay on pre-tassel corn, so on the leaves that are surrounding that tassel, and also in corn that is freshly pollinating. That's because the newly hatched larvae feed in and on the tassel and also on that shed pollen. And our threshold is 5% of the plants with egg masses. It's a cumulative threshold, so if you scouted 2 weeks in a row and got a 2% and then 3% of the plants infested, that would add to 5% and be a threshold. Now, when the plant stage across the field is very narrow, then you typically get a narrow window for egg laying too. So this is an example from Enterkin in 2010, where the pre-tassel stage and pollen shed happened within a fairly narrow time in that field. And the purple bars are the percentage of eggs laid overall on that date. So between the 14th and the 20th here, 90% of the eggs were deposited, that were going to be deposited, were deposited in that 2-week period. And what that means for control is that it improves the spray window. It makes the spray time fairly narrow. If you sprayed right at the end of this period, you would have killed all the larvae that had emerged during the previous week or two. So it makes your product efficacy better. When you have stand variability, perhaps on the other hand, this leads to extended egg laying in the field. There could be areas where there's pre-tassel corn and then another area and another area as that variability works through the field. So instead of a narrow peak like we saw in Enterkin, we can get something like this, where pre-tassel and pollen shed goes on for 3 to 4 weeks and the egg laying is extended. That means you really don't have an optimal spray window. If you spray too early, then you miss these late hatching larvae. If you spray too late, then you miss the larvae that are already down in the ear and can't be reached by insecticide. And this probably accounts for some of the frustration that people feel when they spray a field and they end up with damage anyway. So plant variability can be pretty bad. Back to you, Bruce.
Thanks, Chris. And now we're ready to transition into the specialist roundtable. Joining us now is Dr. Kurt Stenke, MSU Soil Fertility Specialist, as well as Manny Singh, Chris DeFonso, and Aaron Burns. At this point, we would ask you to begin to type in any questions that you have, and we will work with those as our roundtable, and we'll discuss those in just a few moments. Well, good morning, and I see Chris is on. So we'd like to start our roundtable discussion now. Please feel free to type any questions that you have over the course of this webinar into the chat pod, and we'll be trying to answer those. I think to start off, I would like to kind of give especially Kurt a chance. Well, first off, let me back up a little step in there, and that is I'm really happy that these specialists are able to join us today. I know that they're all, or several of them, are teaching classes now, and since the university's gone to entire virtual teaching, I'm sure that's a big change. So thanks again for taking time out of your probably ridiculously hectic schedules right now to be with us this morning. So As they're starting to type some questions in there, I thought maybe we'd start out with Kurt. Kurt is our soil fertility specialist. Wasn't able to join us early this morning because he's teaching, but he is here today to talk a little bit. And I'd like to see kind of some of the things that you have for observations about variability in terms of maybe soil fertility impacts in 2020.
Thanks, Bruce. You know, a couple of things, you know, looking at some of the photos that everyone had up here a couple minutes ago, two things really pop out. And some of these are unique to 2020 and some are more general with regards to land ownership. You know, you look at one of the big things, you know, we look at the number of rented or leased acres today versus owned land. And this becomes a big issue when you talk about long-term soil fertility impacts. So the two things that really pop out that we've seen a lot in 2020 and the last couple of years and in some of the photos you showed this morning would be the two P's. That would be potassium, looking at your critical soil test K levels, and then again pH. You know, and pH really again dictates a lot of what we see in the field year to year. So the first one I'll address, potassium. So your critical potassium levels are going to vary by soil texture. All right. So you can look at, you know, however you variably sample a field, look at the cation exchange capacity across that field. It'll vary to some extent by that specific soil texture. I know, Bruce, some of the pictures you had up there, you know, the soil texture is quite variable across that field, right? So when we look at critical soil test K levels, we're really looking at if you're about that CEC of 5 or 6 and under, that critical level is going to be about 100 parts per million. And that when you're at about that 6 and above or 7 and above, that critical soil test K level is going to be about 120 parts per million. So I know some of the photos we were looking at, there alone lies some of that issue. Now, you know, it's easy to paint that picture as black and white, as in I'm either above or below and I'm fine. But the other issue that we've dealt with extensively the last decade would probably be this variable climate scenario that we're in, right? And when I say that, I'm talking about these 4 to 8 week periods. This year it happened to be mostly July and August, a little bit in June, where we just don't get much rainfall, right? We're looking at an inch and a half, maybe over the course of one of those months. ET rates are you know, probably knocking on the door of 0.2 to 1/4 of an inch a day. So we can lose water relatively quickly. And we got to remember, corn plants don't chew their food, right? They drink their food. So you could have optimal soil test potassium levels, but you will still see potassium deficiency because you don't have enough moisture in that profile, all right, for that potassium to diffuse to that corn root. So in many cases, you can be above that critical STK level and you will still see potassium deficiency show up early, mid, late summer, simply because we turn to these dry weather conditions. The other issue that we've seen a little bit more of this year and a little bit more of in no-till is the impact of soil pH. Now we tend to forget, you know, the overall impact of pH, but pH is still the driver, a big driver of what nutrients are available and the solubility of many of our nutrients. Now, corn can tolerate a soil pH that gets to be quite low. You know, you get down to the 5.4, 5.3, 5.2 and be okay for corn, whereas something like soybeans and some of our other rotational crops are going to suffer extensively. But the key to remember is when you get down to those low 5s, and especially when you get down to below 5 and those high 4s, nutrient availability is impacted from the extent that your aluminum and manganese become much more soluble and much more available, and will impact the availability of other cations. Okay, other cations, I'm talking about your calcium, your magnesium, your potassium, etc. And so because of that increased solubility at that lower pH, we tend to see that show up a little bit more in your poorly buffered soils, so your sandy soils, your coarse textured, low CEC soils. That's where we see that pH and some of that variability really show up. And some of that would be indicated by variability in soil texture, as you can see from a soil map, etc. Some of those conditions with soil pH can be exacerbated in no-till scenarios where a lot of nitrogen over the years has probably been top dressed or applied over the top, not knifed in. So we tend to get this stratification in that top couple of inches of soil where that pH may be drastically different. And then as you get 4 inches, 5 inches down in that root zone, that pH will increase a little bit over time. So a little bit of the variability that we probably saw this year was some of those corn plants kind of stuck in that top 3 to 4 inches of soil. All right, we have elevated levels of aluminum, manganese, maybe a little bit of iron at those lower pH levels in that top couple of inches of soil, which causes some of that yellowing that we see on the surface. We oftentimes think it's automatically sulfur, but if it's at those lower pHs, sulfur availability is impacted at those low pHs also. So it could just be a toxicity issue based on pH. Now, I think we had kind of a perfect storm here this spring when you look at, we had that Mother's Day frost back on around May 13th where we hit 27 degrees. So if you had plants that were maybe in that 2 to 3 leaf stage that were stuck in that top pH stratification layer, you then had that Mother's Day frost, which impacted development, maybe have stunted those pockets of the field a little bit more. Other pockets of the field where those roots can grow out of that top 3 to 4 inches, you will start to see that plant recover. So you probably saw a little bit of stunting in those pockets that then slowly recovered over time. Don't forget, I think it was probably right after Memorial Day weekend, like around May 31, June 1, that we then saw another bit of a cold snap where we were around that 39, 40 degree mark. So again, it's kind of a perfect storm where we saw the stunting early in the season. And then it might have been exacerbated a little bit by some of those mid-May and late May cold snaps that we saw. So again, look at, especially if you have some newly leased or newly rented ground going into 21.
Yeah, I think that's exactly right, Kurt. That's it's a bunch of challenges, that's for sure. Eric, I know we were talking about a soft start or soft break in here, but maybe we could turn it over to Erin just for a second, because I think she might have some comments on pH as well in terms of the herbicides. Erin?
This is Erin Burns. I'm the weed scientist, and pH has a large role in herbicide injury, herbicide degradation. So either extreme, so low pHs or high pHs can impact carryover, and also the microbial community of the soils. So lots of microbes degrade those herbicides, and if they're not as active, we might not see as much degradation. So keeping in mind, looking at your pH when you're doing those soil tests, and then our weed control guide or the herbicide labels will state some of those restrictions based off of your pH.
So Manny, I know we talked a little bit earlier before we did put this, at least when we were in the planning stages of putting this together today, and thought about some of the issues with no-till versus non-no-till and other things. What really kind of stuck with you as being the biggest driver of the variability this season in terms of evenness in corn stands?
Yeah, Bruce. So yeah, I think you pretty much covered, I think, quite a few challenges that we have had this year, right? As I think most of your pictures did allude to a lot of those issues. And again, it depends when the variability is induced, right? You can have issues that are occurring even before planting, right? In terms of compaction issues you were talking about, or even heavy residue in fields that we need to be thinking about, maybe some continuous corn situations, right? Tillage becomes a component in here. And with this push for early planting and the variable planting window, that extreme rainfall events we are getting in the springtime, that can lead to those unfavorable planting conditions, right? I mean, you're talking about your poor soil to seed contact as well. So that all can factor in conditions even before planting, or what is happening even at the time of planting, right? If we have poor field conditions because of these environmental challenges, that can lead to poor plant performance, as you showed in a couple of your pictures too, right? And that optimal planting performance becomes even more critical with these variable planting conditions. Either too wet, we generally think about being too wet, right? But you showed a couple of pictures in there that too dry could be an issue. So optimal seeding depth becomes really important in there. And then there are these herbicide and nutrient management issues that Kurt and Aaron were talking about. And this can all lead to this uneven stand, right? And essentially variability that we are looking at, you can look at two components, right? Either the variability is in terms of differences in plant height, and that we can maybe is manageable. But when we see differences in phenological development, differences in growth stages, that's where some of the yield losses might occur, right? That within those same row units, we are seeing this variable growth. And I think from research, what I've seen is if there are neighboring plants that are at least 2 leaf stages behind, that's where you start seeing some barrenness or some of those stunted ears that will, again, that are a result of a poor pollination because you're dealing with the extended pollination window. And that can even result in some sort of in kernel abortion and even low test weights too, right? And to me, the second important component there is the variability. Either it could be within the rows or you can see these different pockets within the field that have a differing phenological development, right? And to me, that I don't think there's any intra-plant competition going on there. So, but you're looking at essentially 2 or 3 different planting times at that point, right? So we've got to be thinking from that perspective that I would say generally we would think the plants that are ahead we might be thinking that they're going to yield better, but it depends when the phenological development is occurring, right? And when some of these disease and insect pressures are causing, or even when drought stress is occurring. Maybe an area that might be a little bit behind might see benefit because the pollination and the kernel development was occurring in a better or a less stressful environment. So I think there are 2 different aspects here, that within row variability and then like these pockets within the field, and that needs to be managed differently.
Yeah, no, I can understand some of those things, and it certainly seems like we have plenty of pockets and fields if you look around there that do not compete with each other in a negative way. They seem to just kind of maybe along the very edge of those pockets. So I think that's pretty good.
Yep.
So Bruce, we had a couple questions. We were talking about pH earlier. So I'm guessing these are both for Kurt. I'll give you both of them. What's the best soil test? So if you're looking at pH, are you doing 5-acre or 1-acre grids? That's one question. And the second question would be, should liming practices be modified when we have no-till?
Thanks, Eric. Both of those are good questions. You know, the second one, should liming practices be modified for no-till? Typically, if you're in a no-till scenario, you really want to regular monitor the pH in that top 3 to 4 inches of soil. So, you know, typically your typical soil sampling depth will be about 0 to 8 inches. But in no-till, oftentimes you want to keep an eye because that pH stratification is so important on nutrient solubility. You really want to look at that 3 to 4 inches, that top 3 to 4 inches in that no-till scenario. The other question about grid size, you know, a lot of that depends on your budget, right? And how high density of a soil sample you want to take. So, you know, the old standby, you know, make sure your samples don't represent any more than 25-acre grids. Then typically, if you have these niche areas of the field, might be topography issues, might be soil texture issues, could be a wet hole in the field, things like that, and you want to do a higher density sampling, that's fine. Typically then we're talking about maybe, you know, around 2.5-acre grids. And if you really have a, you know, some room in your budget, then you can get down to the 1-acre grids. But typically we're talking, you know, that higher density sampling is typically about 2.5-acre grids or so. And a few growers occasionally will go down to that 1-acre grid size.
Excellent. And those are both excellent questions. Eric, I see we're at 10:55. Is this question if you want to do your soft break and then we'll come back with more questions?
Yeah, so this is a great discussion, and I definitely don't want to cut it off. And we will come back for more Q&A, but I know that some of you probably need to get going at 11 o'clock for other appointments. So I just want to go over a few logistics before we get back to the questions. During a morning field day like this, there's really no way that we could address the numerous challenges that corn farmers need to manage, but we do want to make sure that we make you aware of valuable resources or through the extension. The Field Crops team website is a one-stop shop for answers to many of your questions. Here you can find recordings from past virtual breakfast and webinar series sessions, as well as podcasts and links to timely articles and upcoming events. Clicking on the Experts tab will take you to a list of campus specialists in many different disciplines, as well as field-based staff who cover your region with their contact information. The farm management team has so many resources, I don't even have time to read through all the different topics they address. You'll definitely want to visit their site and browse around. You'll find answers to questions you didn't even know you had. Aaron already mentioned the MSU Weeds website contains an electronic version of the Weed Control Guide, which is updated annually. Several articles, bulletins, and videos on all the weeds that are currently challenging crop farmers. You can also visit the MSU Weed Science YouTube channel, where all their videos are conveniently compiled. You heard a bit about irrigation research and outreach here at MSU. We also have resources for those with too much water. At this website, you'll find information and tools to address your drainage needs. Nematodes are one type of pest we did not have time to address today, but you can find out more about the different pest species and current research at this website. All right, so please take a few minutes to give us your feedback and a brief survey about today's virtual field day. We learn new things each time we host these virtual events, and we want to keep improving our programming for you. The link for the survey is being posted in the chat box. Please copy and paste that into a browser window on your computer or a mobile device. For those of you who are joining over the phone with voice only, you can send a text message to Megan Honke at 989-666-3773, and she will text you the link. I'll give you that number again in a minute. At the end of the survey, you'll be asked whether you would like to obtain RUP and CCA credits for today's live program. So for those of you who are interested in earning credits, be sure to go all the way through to the end of the survey for those codes. So again, if you're voice only, that number again to text Megan is 989-666-3773. So thank you again for joining us today. We hope you found lots of good information you can use in your farms and in your businesses. Bruce, I'm going to hand things back over to you for more Q&A.
Absolutely. Thank you. Appreciate it, Eric. So let's go back to our discussions a little bit in there. I know when we think about some of the discussion earlier, and also we'd like to invite Marty and the rest of his crew and Lyndon to talk a little bit about irrigation as we go into more general questions. One of the things that Manny was talking about in his talk was looking at earlier planting or utilizing that growing window or that growing season window to its fullest extent. And I guess maybe that's kind of a good question for all of our specialists. Exactly what implications do you think that has for either fertility or weed control or insect control? And what are some of the benefits and what are some of the drawbacks? So I'll just throw it out there. Any of the specialists want to pick it up? Let's go ahead and do it.
This is Erin. I could address that on the herbicide side. So if you're planting earlier, our temperatures are most likely going to be a little bit cooler, and that has implications on how effective some of our burn down herbicides work. So on many of those herbicide labels, you'll see that the statement is they need to be applied to actively growing weeds. And so when it's cold, the weeds aren't growing, and you might not see the control you'd expect if it was a little bit warmer. So usually we recommend about 50 degrees is the cutoff that you want to be making some of those applications. And if we did have a snap hard frost like we did this past spring, you'll want to make sure you wait a few days to apply those herbicides, because you need to give those weeds a chance to regrow. So those would be the implications on weed control.
And Bruce, I just want to add, I think again, the idea is that we need to be matching our maturity selection with the growing season that is in front of us based on when we are able to plant, right? So I think in corn, what we are finding out is that uniform growth and development is important, right? We are seeing this variability, and part of those is that early planting and the non-ideal field condition. So I think, again, that's just a comment I think I was talking about, again, not only pushing for early planting, but optimizing our maturity selection with when we are able to plant, right?
Yep.
Because I think unlike soybean, again, in soybeans, we are finding out that, okay, you can maybe able to push to early planting. And even if you have uneven stand, as long as it's not really non-uniform and you have these big holes, the soybean plant is able to compensate for that low plant stand. But corn does not behave like that, right? So again, we got to be careful there.
Chris, does early planting have any implications for insect controls that you have worried about?
Yeah, and it's kind of all over the board. I mean, if you're going to plant early and if you're going into cold soils, that just gives more time for the seed to sit there, and things that eat the seed or the little plant as it's coming out of the ground have more time to be active. So wireworms, grubs, those sorts of things. And then by planting a little bit later, when things kind of pop out of the ground, then that plant is able to outgrow that insect. There are also certain things like some of the grubs finish before the others. So for instance, Asiatic garden beetle would emerge. You could, in theory, plant after it's already pupated, whereas Japanese beetle will go a little bit longer. But that requires a knowledge of what's kind of going on in the field, and I don't think that people will delay planting in order to delay damage from that insect. And then, as I talked, once the planting is set up, then later in the season, something that was earlier or late planted could be attacked by an insect or avoided by an insect. So for instance, I talked about Wister bean cutworm, and it's how its flight lines up with pre-tassel corn oftentimes. And it's not just your individual field, it could be the whole landscape. So if you're the early field in the whole landscape, and that's the most attractive place, you may get kind of dumped on by a particular insect. Something like corn borer too. First generation corn borer will go for the tallest corn. So that would be the earlier planted, where the second generation is going to go for what's still pollinating. And maybe that's a later planted field. So it seems like for insects earlier and late planting, it's sort of all over the place, and one insect can be worse and one insect can be better.
Thanks, Chris. So Kurt, what's the verdict on going earlier with corn in terms of fertility? What are the kind of the key things to keep in mind?
One thing that will never change is make sure you look at that soil test when you're looking at what to put in that starter fertilizer. So typically, again, look at your P&K levels. If they're above critical in that maintenance plateau range, you may not have to include those in that starter fertilizer. But again, those 5 magic words that my programs looked at a lot over the better part of the last decade is start right to finish well, right? And so we've seen that benefit to starter N many times over. Where we've seen it the most is probably in these drier spring and early summer periods, where we can drive a little bit of corn growth early. And the thing to keep in mind with starter fertilizer is, do you know more 40 days into the growing season or at planting time? And I think most of us would agree you know more probably about 40 days into the growing season. So what that starter fertilizer does is it lets you adjust, right? You can take a look at the environment. You can take a look at your yield potential. Is it the same as it was when you planted? Is it higher? Is it lower? And then you can adjust that side dress end rate, and then potentially save a few dollars on your nitrogen investment, or perhaps go whole hog if growing conditions warrant that, to really meet whatever yield potential that specific field or grower may have in mind. So again, that start right to finish well can pay dividends. Just make sure with what you put in that starter, you look at that specific soil test.
Yeah, that's undoubtedly true, Kurt. So I guess my next question to all of you is Manny's observation, and that that we have wetter conditions early in the spring, what's likely to move that we put on, and under what conditions?
As far as spring applications, are you looking at fall applications?
Kind of a little bit of both, a little bit of combination. If we do put stuff on in the fall, I assume we're more vulnerable depending on soil type, right?
Yes, correct. So, one of those, the popular topics or popular nutrients that many people are talking about and continue to talk about is probably sulfur, right? So when you look at sulfate sulfur, remember it is an anion, right? So it's negatively charged and can move and leach through that soil quite readily with some of those big rainfalls. So that would be one aspect to look at. We don't really have a good soil S test, so we can't gauge whether to apply or not apply simply based on that soil S test. So it has to go with a little bit more on what we've seen in the past and a little bit with tissue testing probably early on in the season. And so that critical level for most crops would be around 0.25% sulfur. That's a good gauge. When you're above that, you usually are sufficient. When you're below that, you're usually deficient. It'll vary a little bit by some of your higher S using crops, something like alfalfa and onions are a little bit more higher S using, so you can't go off that 0.25%. So that would be one to look at. Then the other one to look at would probably be the leaching and or denitrification of your nitrogen application with some of those maybe early pre-plant applications. And it becomes a question of, if N does move, can we then reach it? And then you got to keep a good eye on that field in season to scout and see if you're seeing some of that N deficiency. So on your coarse textured soils, we're much more concerned with leaching. On our fine textured soils, we're much more concerned with denitrification. So if you're well tiled or have recently upgraded the pattern of your tile, maybe cut those 33s in half or those 66 strings in half, you're much less prone to denitrification. And so again, it's the shoulder seasons, right? The shoulder seasons that we're talking about with regards to N application. And those shoulder seasons are those extremely wet and those extremely dry spring seasons. And interestingly enough, we've been about, 50-50 mix of extremely wet and extremely dry here in Michigan with regards to that early spring pattern over the last decade or so. So again, those PPI applications can be very effective. They tend to get a little bit of a bad rap in the big picture, but they can be very effective. But it's, again, those extremely wet, we're talking about those 8-inch Aprils or 8-inch Mays of precipitation, or those extremely dry seasons where those PPIs will tend to suffer a little bit more than your at plant and applications.
Yeah, absolutely. So what about you guys? Aaron, what would be the major concern with wet spring and heavy rainfall and with the weed control?
I think two roles. So we have, herbicide injury implications. If it's cool and wet for a long period of time, that's when we might see some of that injury from those pre-emergence residual herbicides. And then also just if you get a big rain after you apply that herbicide, depending on your soil type and what herbicide you applied, some of that could move out of the upper 2 inches of soil, which is really where we see most of our weeds germinating from in the early spring. So then you wouldn't get as good of weed control as you might expect. So we have some resources at the end of the weed guide that kind of talk about leaching potential of herbicides, but also feel free to call me or shoot me an email. I've worked with growers before looking at what, exactly what they applied and what might still be around and what we're going to have to scout for, particularly with some weed breaks, and then what we can apply after some of those big rain events.
Okay, thank you. Chris, I'm coming for you next. So I guess I was just curious if we could drown any of those critters out there that were going to be potentially causing us issues.
Well, if you drown them, that also means you have standing water, and that's not helpful either. So I don't think we can rely on drowning for most things. But the cool wet weather, a lot of rain, it just slows down emergence and gives more time for these insects to feed. It also probably, things like slugs would be of greater concern. But probably the biggest thing is just it's the weeds that the poor weed control that Aaron would be challenged with, then for me, that's when black cutworm comes into these weedy fields, or true armyworm. So when I look at that, I think it's more of a weed problem that then leads to an insect issue a month or two later.
Yeah, I think that's what we've seen down, at least in this part of the world, and there too. Hey, Marty.
Bruce, I've got a couple questions for you from the Q&A in the chat.
Perfect.
Let's see. I think the first one, we'll go back to Marty. Marty, does tar spot affect corn grain feed quality?
So no, I guess in general. So, I mean, it will affect test weight. So yes, yes and no, right? There's no mycotoxins though, right? So I think that's the big issue we think about with corn diseases, especially like gibberella, ear rot, and you know, Chris was talking about Western mint cutworm, opening things up, letting gibberella or fusarium in there, and that, you know, that results in mycotoxin very often as well. So that's obviously a big concern. But there's no mycotoxin associated with tar spot, so no real impact on that side of things. We had a quick look-see at some silage a couple of years ago now, and the issue there is basically just premature plant senescence and the plants cannibalizing themselves and the fungus probably feeding on the sugars and things too. So tar spot will reduce silage quality in terms of energy, and basically the fungus is consuming some of those digestible components and leaving you the non-digestible that you don't want to increase in your feed ration, right? So, yeah, somewhat in terms of quality, but thankfully no mycotoxins.
So Marty, it can make harvest real interesting though, can it?
Right, and you have some personal experience with that, right? Would you like to talk about that?
Yeah, I mean, the stuff dries out real fast. So if you're looking at like corn silage, it can turn on a dime, and especially in drier weather in there. So you have to really watch that. It's been a real harvest challenge down here. And of course, our field corn, if the stalks get weak, and we have problems with lodging later in the season too. So that's another issue just for that doesn't get cut for silage. But it's definitely it makes it interesting to do the harvest operations.
Right.
Along those same lines, Phil Cates is one of our forage educators, mentioned that starch can be affected, less of the starch due to the amount of grain fill. Also, just want to let everybody know that folks have been addressing some of the questions in the chat box. So if you don't have your chat box open, you might just want to open that up. I know Chris DeFonso has been writing like a house of fire in there, so feel free to go through and read some of those questions and her answers to those back and forth. I've got a question for, I'll say, Kurt. Kurt, what's the better application for nitrogen on corn? Would you say top dressing treated urea or ESN, or side dressing 28% with a stabilizer?
Thanks, Eric. That's a great question. So, you're looking at a couple different modes of action on some of your enhanced efficiency fertilizers there. One thing is that we need to remember is that whichever mode of action or whichever product you use, you have to have N-loss conditions to see a response to those products. All right. Under favorable conditions, favorable growing conditions, there's no reason to see a response to a lot of those products. So then it becomes a matter of, come springtime, trying to predict what this year is going to look like, and each year is different. And we could sit here and talk about that forever. So, but the first question, or the first answer, is you got to have N-loss conditions. Beyond that, if you're looking at something like, an ESN urea combo, again, remember that ESN product is a controlled release fertilizer, whereas the other half of that question, you talked about side dressing with a stabilizer, right? And it really depends what type of stabilizer you use. Are you talking about a urease inhibitor or a nitrification inhibitor? And when do you plan on side dressing? So if you go with something like a urease inhibitor, you got to make sure that it's a surface application. Otherwise, there's very little reason to use a urease inhibitor if it's subsurface injected or for some reason broadcast applied and incorporated. The other stabilizer category you'd be looking at is probably a nitrification inhibitor, all right? And that keeps that N in that ammonium state for a longer period of time. before transforming to nitrate. And the answer there is, when are you side dressing? So the later you side dress, the less likely you will see a response to that nitrification inhibitor. So if you're side dressing your typical V4 to V6, or maybe even a little later towards V8, or maybe the late vegetative stages, there's probably very little reason to use a nitrification inhibitor, because the probability of seeing a response just isn't going to be there. Because once you hit that V6 to V8 corn growth stage, that corn really takes up that end really quickly. So that would probably be geared more towards an early side dress, or even an earlier maybe at plant application, something like that. When we've gone with pre-plant granulars, you're looking at your urea and ESN combinations. We've typically gone with something like a 75-25 mix of those products, and have seen some pretty good responses. You want to keep some urea in there so you get a little bit of soluble N early in the season, and then you can have that controlled release product that can release later in the season. So there's a couple moving parts there. When are you side dressing, or are you going with an all pre-plant application? And then just make sure that a controlled release product and a urease inhibitor and a nitrification inhibitor are completely different modes of action with regards to enhanced efficiency products.
Kurt, in that same vein, I guess, have you seen any advantages to like location within the closeness to the plants when you're trying to do side dressing?
Bruce, you talking, something different from like a 2x2?
Yeah, well, I guess that's kind of what I'm thinking is, so even some of these are kind of a directed side dress where you're putting a little closer to the plants and the rows are split versus just one down the middle. Has there been anything that you've seen that's made a major difference?
Yep, so when we've looked at side dressing, maybe with something like a wide drop applicator or something like that, where it drops that end closer to the plant, we have not seen yield benefits to something like that, especially under dry conditions, as compared to something like a Coulter Inject. The Coulter Inject still is a good old workhorse. It does very well, but the problem is it's probably, yes, a little slower than some of the alternatives that are out there. So, you got drop nozzles, you got wide drop, you got Coulter Inject, you got all sorts of types of infrastructure you can use to apply end now. But applying that end closer to the plant, we really haven't seen many benefits as compared to something like your typical Coulter Inject. One question that has come up more recently, we've talked a lot about 2x2 over the last number of years. And so a lot of producers are looking at 2x2x2. Right? So you're looking at 2 inches to the side or two inches to the side, to each side of the corn row. And, we have to realize that the corn plant just does not take up end from an individual side and keep it to that side of the plant. So we really haven't seen much benefit to a 2x2x2 application compared to something like a 2x2 application.
Good. Eric,
So,
go ahead.
I've got one question, and Chris, I know you've handled this a couple different times in the chat, and we've got another one in the Q&A. So everyone's really interested in how do you control Asiatic garden beetles. So maybe you could just kind of summarize everything that you've been talking about in the chat.
So Asiatic garden beetle is a tough one. One thing we do know is that most of the fields that are affected kind of the year after year after year in Southwest and Southeast Michigan are these kind of sandier fields that have at least 80% sand. So if you have fields like that, that's where you would be looking for Asiatic garden beetle. Now this time of the year, the grubs would be there. They would have come from eggs that were laid like July, and the second or third stage grubs, the third, the biggest, would actually be present. I assume that tillage would help, but a lot of guys don't want to do tillage, but certainly they're under the ground. It would kind of break up their overwintering. You could go out and look now, but most people are busy, or look in the spring. If I had fields that I knew were my Asiatic garden beetle field, I think I would plant them last, because Asiatic garden beetle pupates fairly early. And if you can, the less time your seed has in contact with that grub, the better. The other thing is we do have some insecticide choices. We don't have good replicated data yet. The low rate of the seed treatments are garbage on this pest. They just eat through it. Higher rates may work a little bit better, but they don't seem to be the total answer. We've had plots with a Counter and Aztec granular, as well as Ampex, which is a liquid clothianidin, that you can see a reduction in adult emergence. There's also a product called Index, which is a combo of chlorothoxyphos, an old organophosphate, and bifenthrin, and it's the chlorothoxyphos component that seems to smoke these grubs in the lab. My colleagues at Ohio State have done some work in lab tests in cups. Now, how does that translate to the field? We don't know. So 2 years ago, 2019, we had some plots out, but we didn't hit the right field. You know, there was kind of moderate infestation, and this spring, we were looking for really heavily infested fields to run in some plots to answer these questions, but then COVID hit and we weren't allowed to go out and do that. So I'm still thinking about if you're in Southeast and Southwest Michigan and you have problems and we can get out and sample fields, there might be a way to run in like with and without sort of plots and address that question. It's usually not stand loss, as Bruce knows down in his area. It's more of a almost subtle differences in plant height, and we just haven't hit the right place to get these replicated trials in. Bruce, do you have anything to add as far as management? I mean, you can't add enough cranes to a field to get rid of a garden beetle, you know, to eat them. So
Yeah.
you know, it's a tough one. And it's unique to our area too. It's a Great Lakes sort of problem.
Yeah, I always wonder if that's part of the draw for sandtail cranes, just get them something to chew on long enough for that corn to come up to become more tasty. So
yeah,
I guess the only other thing I would add, Chris, is do you expect to see more challenges in fields that are beans this year, or are in continuous corn? Is there a dynamic difference between whether something was rotated or not?
Most of the infested fields, most of the bad corn fields came after soybean or potato down in your area where there was a dense canopy, but that doesn't always hold. So every time I think I know something, then someone tells me, no, that isn't the case in their area. And it's like, oh, it's not, it's not right. So, but usually it's after a thick canopy of something.
Yeah,
we've seen it in alfalfa too, and I presume if that alfalfa field got worked up and you planted after it, then you'd have problems there too. And it's not like soybeans are innocent. I've driven across the border into Indiana and seen an occasional field where the soybean stand was thin. Now it was a crappy, it was kind of that sandy top of the knoll kind of thing where it wasn't great anyway, but there was AGB there as well. So we've seen it in many crops.
Yeah, I always think about that when we were looking at alfalfa stands that look pretty unthrifty on some of those sands too. It's kind of like the middle side of that hillside that shouldn't be bad that suddenly looks not so good. It's
usually not your best productive field, let's put it that way. So
yeah, that's true. Eric, is there any other questions that we need to worry about here?
No, I have not seen anything come in the chat or in the Q&A. So this could be a good time to wrap up. If you have questions that come to you, feel free to reach out to me or one of the specialists. I can forward your questions on to them. I just want to, thank our partners once again from the Corn Marketing Program of Michigan for their support, especially Kristen Poley for helping today's program run smoothly. And thanks to all of you for attending and providing us with your good questions, good discussion. I hope to see you this winter during our various winter meetings. Have a great day.